Practice 6. Purification of a PCR product from an agarose gel.
Introduction. The DNA purification is one of the most important methods in biotechnology because it's a first step to make a new experimentation and it will determine the success or the failure. Two reasons that DNA purification will help you are: 1. You can extract ample amounts of the genomic DNA from a specific source that is needed, because sometimes we don't want all the DNA segments. And second, it reduces the amount of contaminants that can disturb the final results in the research and reduce the life of the samples. There are some examples of DNA purification methods, and they have some advantages and disadvantages than the others in specific cases. For example, there is electrolysis, and some advantages of this method are acid-base centrifugation, heating, vortexing, and allows small manipulation samples. In contrast, one disadvantage is the usage of expensive apparatus and consumables membranes. In the practice was used to the Pure Link Gel Extraction Kit K21K from in vitrogen, which is quickly and efficient method of DNA purification from agarose or TBE gel with many percentages.
The protocol used on this practice is the following. First of all, make electrophoresis to 0.8% of agarose and 0.3 x concentration for Hellred, and for 1 hour with 1x TAE gel. Display the gel in the trans-illuminator and cut the band with a knife. Place the fragment in a microtube and weight it. Add 790 microliters of L3 buffer. Make an incubation at 50 Celsius degrees for 10 minutes, mixing by inversion every 3 minutes. Put the mixture in the column. Make a centrifugation at 12,000 revolutions per minute for a minute and discard the filter. Add 500 microliters of W1 buffer. Make a centrifugation at 12,000 revolutions per minute for 3 minutes and discard the filtrate. Add 50 microliters of E5 buffer and incubate for 1 minute at room temperature. Make another centrifugation at 12,000 revolutions per minute, in this case for 1 minute. Finally, was necessary to make a quantification using a spectrophotometer and electrophoresis. To use the spectrophotometer was necessary to use 10 microliters of DNA sample and 990 microliters of molecular biology grade water, and the measurements were on 280 and 216 nanometers, and the electrophoresis conditions were 0.8% of agarose gel with 110 volts for 60 minutes.
Results. The results obtained of the extraction protocol are as follows. By making a gel electrophoresis, the desired band was selected and extracted. A well-defined and thick band was removed from the gel, which means that there was sufficient genetic material. The Pure Link kit was used for further purification by means of a silica column. After the procedure was done, the 260-280 index was obtained, and the result was 0.3675. Discussion. Although the selected band possessed desirable characteristics and it was estimated that the amount of DNA present on it was sufficient, when the sample was analyzed with the spectrophotometer obtaining the 260-280 index, it's shown to be off the charts with a value of 0.3675. Since the ideal value for this is around 1.7 to 2, this meant that there was an important contamination, and a value of 0.5 means that the sample is not suitable for further use due to contamination of either proteins, carbohydrates, or other compounds. However, during the experiment, the origins of the sample were well known, meaning that it was only DNA, so the contamination with proteins and carbohydrates must be discharged. Therefore, the contaminating must have been with phenol. that was not washed away correctly during the procedure of purification. It would be highly recommended to check in the index 260-230 to verify if it was the phenol that contaminated the sample.
Conclusion. To maintain the certainty that a practice was successful, there were some important parts of the protocol. For example, in first place, there is the quality of the DNA previously made and the conservation. In second part, the usage of the agarose gel at 0.8% and the usage of TEA by its lower concentration of salts were essential in the electrophoresis. The quantification in the spectrophotometer in first sight was too high, but after one additional dilution was correct in the standard.